Now I'd like to introduce today's speaker, Dr. Mauro Rebelo, who is at the Institute of Biophysics at the Federal University of Rio de Janeiro. Mauro is the head of the Environmental Molecular Biology Laboratory. He has interest in marine ecology, molecular biology, and ecotoxicology. He has projects on pollutant toxicity mechanisms, invertebrate genomics, and metagenomics of aquatic environments. He's also an entrepreneur and the founder of the company BioBureau. BioBureau focuses on biodiversity identification as well as the development of genetically modified organisms which may be used to control invasive species, biomining, and for the production of compounds of significant commercial value. Mauro, thanks for taking the time to join us today and to share your work. We really appreciate it. So at this point, I'm going to turn things over to you.
Great pleasure. Thank you for the invitation and the opportunity to share with you all the work we are doing with the Golden Mussel, which is, today I'm going to go through all these points that you have on the screen, why we should control the Golden Mussel, why we are using gene drive and not other traditional or biotech technologies. I will talk a little bit about the genetic program on the Golden Mussel. That's kind of a requirement to do what we are doing, as well as the challenge of raising an invasive species in the lab, which should not be, but it is. Some of the modeling we've been doing to assist us in the development of the gene drive, some aspects of the gene drive itself, the challenge ahead, and I will end showing you a beautiful picture of our team. And to start, maybe I would like to show to you this short animation video that tells the story of the reason why we should control these species.
Hi there, my name is Marcela and I'm a PhD student at the Biophysics Institute at the Federal University of Rio De Janeiro in Brazil. I'm going to present to you my PhD project, that is the genome sequencing of the Golden Mussel. Well, you all know what a mussel is. Usually they are very delicious to eat. But this one I work with, that is about 1 centimeter in size, we cannot eat, and it's causing lots of troubles. It came from China in ballast water, the water used to balance ships when they are empty. It arrived in Argentina in 1991 and rapidly spread through South American continental waters, reaching the wetlands of Pantanal, very close to the Amazon River basin. This research is part of a bigger project to avoid golden mussel reaching Amazonian waters. You know, these mussels took the saying grow and reproduce too seriously. They grow side by side, one under the other, they grow, grow, grow. In 1 year, their densities go from 15 mussels per meter square to 150,000 mussels per meter square. Like this, they clog pipelines of power plants, attach to ships' hulls, trees, and even to other native bivalves. They are unable to open their valves to breathe and reproduce and end up dying. But that's not the worst part. Without natural predators, they are well adapted to the tropical climate and now they only do 3 things: eat, sleep, and mate. Not necessarily in that order. They mate much more than they sleep and they eat even more. They eat so much, filtering the water, that make the algae disappear. Fish eat mussels too, but they eat so much that they end up dying of indigestion. Scientists call it an ecosystem engineer, because it changed the invaded environment, decreasing biodiversity. Actually, we do not know enough about this mussel. We know it can attach to any surface through the byssus, produced by the mussel's foot gland. But how this foot gland can do that? This is the kind of question we are eager to answer. We want to study its DNA and genes to be able to understand the mussel better, but that costs a lot of money, and that's why we need your help.
Okay, so this is the video we produced for a crowdfunding campaign in 2013 that helped us raise $20,000 at the time and allowed us to start the genetic program of the Golden Mussel. Even though we were already working with transcriptomics and trying to identify genes, but it was a kind of kickoff of the genetic program, this crowdfund campaign. So you can see here some very impressive images. This is the armado, one of the fishes of the Paraná River Basin, which is the river that ends up in Argentina at La Plata River. So if you open the stomach, you can see all the mussels inside. And if you open the intestines and you take those mussels out of the intestine and put them on the water, they are still alive and they are going to open and start to filter. It's very, very impressive. This image here is aquaculture. It's a fish net for aquaculture. And as you can see, the mussel, they clog completely the net. So that's how it starts with this image here on your left side. They start to grow and they grow, and they clog completely the fish net. It's another short video from last year. Now it's really hard to get them out of the net. So what they do is, just leave the nets on the sun, under the sun, for a couple of days, like 4 or 5 days, depending on the amount of sun, and the mussels start to die and to detach, but they go back into the water. The smell is terrible. And this is one of the problems for some of the economical activities in the reservoir. Of course, you have the main impact is in the hydroelectric power plants. So this is an image of one of the grids that protects the water capitation to go to the turbine. And it's simply so many mussels, so many. The problem is very similar to what a lot of companies, of utility companies in the US suffer with the zebra mussel or the quagga mussel. And the reason why we started a biotech approach to this mussel, it was simply because traditional approaches never work. They don't work. Simply putting poison on the water, the same way that we do in agriculture to control invasive species in agriculture, it doesn't work. The poison gets diluted. It's very difficult to reach to the target species. You just have to put a lot more poison than you normally do in the terrestrial environment, and it doesn't work. So this is one of the articles that we publish in my ecotoxicologist past, showing that sometimes these microencapsulated biocides that get concentrated just inside the mussel because they filtrate the particles, they can show, they have more potential than other diluted pesticides, but they only work inside the power plants, the pipes, and they cannot work in the environment. So, and you can, if you can control them in the inside the power plants, it's just a matter of keep controlling them, because the larvae, they keep coming from the environment. So we start, okay, this is going to be endless. So we need to find a way to control. We need a different approach to control the mussel. And it was in 2016 when I was presenting actually the successful case of the crowdfunding to to start a biotech approach. I was in an event in Italy, and there was also some of the people from Crisanti group, and the article on Nature Biotechnology on the CRISPR for Anopheles had just came out, and I started to talk to them and said, wow, maybe mussels and mosquitoes, maybe we could apply this technology to the mussel also. And at the time, our best guess was to develop some kind of RNA interference. That was full of challenges, not only to, really to encapsulate the RNA, to use it in the environment. Imagine putting it in the water to go inside the pipes in a hydroelectric power plant, but how to get the mussel, what was going to be the delivery system for these particles to reach the mussel. So we were not convinced it would work at all, but it was our best guess at the time, and we were working in this direction. Still, people at Minnesota University, they actually believe the RNA interference approach that has a higher probability of success over gene drive for zebra mussel. So they are developing RNA interference approach for the zebra mussel there. But I thought it was so beautiful, the mechanism of the gene drive, that I sat with Andrea Crisanti, and we both agreed that yes, there was no reason why it could not work on the golden mussel. So we started to imagine how it would look like, and we published in 2018 preprint explaining our rationale behind the strategy of the golden mussel gene drive, all the steps of the TRL, of the technology readiness level scale, how we were imagining the technology development program, and how we, and in all the safety measures that we're willing to apply, which is basically taking all the best practices that were being created at the time and being published, and try to apply this to the mussel. This is, I will skip this video, but you all have access to this presentation and can see it's a 1-minute video explaining how we would modify the mussel in the laboratory and how it could lead to the population control. So, as I explained, the biggest challenge that we had at the time, and that we still have, is that the golden mussel is not a traditional biomedical research model. So lack of information is our worst enemy. And we don't have years and years of research and published information about the species. So what we understood at the time, that the way to leapfrog that lack of information was to develop a genetic program and to sequence the genome in a way that would allow us to get all the biological information from other species and kind of use it to better understand the mussel and how to, and give us the basis to modify it. So the first transcriptome we published it in 2014, then the mitochondrial genome in 2015, then a draft genome in 2018. That's where we used the resources of the crowdfunding, but it was not enough. For you to have an idea, when we started the project, which means when we got funded by the hydroelectric power plants to start developing the gene drive, we said, okay, we have the genome. Let's try to see the difference between the males and the females. We got, so we extracted DNA from the males and from the females and sequenced it. And to our surprise, there was no difference. So we didn't have a karyotype for the mussel at the time. And it was just starting to show up evidence that these animals may not have sexual chromosomes. So again, even if you have the genome, you don't have all the answers. So we kept working on a higher quality genome, and we did now a platinum genome. The preprint was published last year. The article has been submitted to Giga Science and has went through the first round of review. We are confident it's going to be published this year. And you can see that now we have a karyotype here. They have 15 chromosomes, and we have an assembly that like with 99.9% of the genes distributed into 15 scaffolds. So we have a high quality chromosomal level genome for the mussel now. And this new genome allowed us to identify the target gene for the gene drive, which is the DMRT1 gene that I'm going to talk about shortly. So the second big challenge was to produce. So we somehow assumed, even though a lot of groups in Brazil have tried to reproduce the mussel in the lab without a lot of success, some groups in Argentina, they managed to reproduce the mussel there, and we were confident it was going to be simple, and it ended up being the biggest challenge of the first phase of the project. We simply could not reproduce the mussels there, the mussels here. Now, with a dedicated team of people with experience in raising commercial marine mussels, we managed to have mussels reproduced and growing in the lab, growing through the whole metamorphosis and becoming adults. This is a picture of when they were 3 months old. They are now 5 months old. That's how they look like. And well, like you can see a smile in my face because I'm so proud of like I have two children, but I'm also so so proud of these small mussels. It was a big effort from our team that managed not only to reproduce a few of them, but we managed to scale up the production of embryos in the lab. That was a requirement for us to be able to do all the transformations that we need to test the gene drive. So this graph shows the number of embryos and mussels over last year. That's when we managed to have all these successes in reproduction and raising the mussels in the lab. Okay, a very important aspect, again, connected with the fact that we don't have a lot of information about the mussel, is even the degree of infestation, the severity of the infestation. So this first image that you're seeing is a power plant reservoir in São Paulo. It's the Três Irmãos reservoir. It's a huge reservoir, 800 kilometers square, and we do some environmental DNA research in this reservoir, but we were using different methods. The project was using different methods to try to estimate the infestation there. So this image, this data was collected with traditional methods like diving, throwing quadrants and counting, but also with image and artificial intelligence, using artificial intelligence to identify and count the number of mussels in the images, which allowed us to make a map of the infestation and to understand the key points of infestation that you can see in this second image here. So below you see the same reservoir that you see in the top, the Três Irmãos, but this is part of the Paraná River Basin and the Tietê River Basin. And you can see that there are some hotspots of infestation, and these are transduction channels, like channels to send artificial channels to send transposition channels to send water from one reservoir to the other, and the aquaculture farms. So yes, the mussel can attach anywhere, but it prefers to attach in artificial structures. And it's clear to us now that the aquaculture farms, they are a victim, but also a perpetrator of the mussel infestation. This third graph is, so these two graphs are a model of the gene-drive dispersion in the population, estimated using LIMS, the same model system used to estimate the dispersion of the gene drive in the mosquito population, but of course with a lot of limitations, because we had to adapt to the mussel system that, for example, has external fecundation, not internal. It's simply so different. So we are all over LIMS now. We are trying to adapt it to the mussel system, but these are the initial results that we have. So here you can see that in our best case scenario, if we could have some kind of pre-gene drive traditional control, we could aim to control the population in less than 10 years, without this control in about 10 years. And here in the top, it's a simulation of how, in the quadrants, depending on how many mussels you release, what would be the rate of spread to the other mussels. So these the yellow dots are mussels or like invasive mussels. The blue dots are the genetically modified gene drives and how it spreads through time. These are unpublished data so far. So if people are not familiar with the gene drive, we decided to make our own infographic, because we're just showing mosquitoes, and as you can see, the mussel, it's much more beautiful, right? So it's a simple infographic to try to explain, mostly to lay people, and to the main target for our communication now are the hydroelectric power plants, not only because they are funding the project, but also because we need to work with them towards developing these solutions. So with operational people, maintenance people, the mechanical engineers that work with the problem on a daily basis. So we first explain Mendelian heritage, how it works, and how a modification could be diluted over time, over the generations, and eventually disappear. And on the other hand, if we have a super Mendelian inheritance, that would mean concentrating the modification over the generations. And eventually, when we have when the modified genes, when the modified muscles start to reproduce among themselves, you have the sterile muscles that cannot reproduce and end up controlling the population. So this image is, I'm sorry, it's in Portuguese, but I'm going to walk you through it. It's the construct of the, so a graphic representation of the gene drive construct that we are using with its components. So the main, I believe the main information here is the target gene, which is the double sex male abnormal related transcription factor, DMRT1, which is a gene very important for the gametes, and without which the gametes, that's our strong assumption, cannot reproduce. And this is the same gene used in the mosquito. We did a lot of work led by our bioinformatician João to identify the genes in this new genome and get to a transcript that we believe is the one. So we synthesized this construct also using the VASA promoter, and that was another huge bioinformatics effort to identify this promoter in the genome of the mussel, because we need a promoter that is activated just through the gametogenesis, right? To have a mussel that is modified, but that can still reproduce, but creates that all its, even though it reproduces all its gametes, not only 50% of its gametes carries the modification. So in this construct, we have the VASA promoter, we have the DMRT gene, we have, of course, a green fluorescence protein reporter to give us evidence that the gene was that the construct was incorporated in the cell, and the Cas9 that creates the gene drive. So here you have two pictures, two evidence of course, it was a big challenge to put this big construct inside the cells. So we managed to transfer the construct to make the transfection of the construct inside embryos, but those embryos didn't survive yet. So the survival rate of the genetically modified embryos is very low, and because we were still scaling up the embryo production, we still didn't manage to transfect as many embryos as we need to be able to have the genetically modified adults that came from the genetically modified embryos. But this second image was the expression of the gene in the gonads of adult mussels that were injected with the construct. With the larvae and the embryos, the only way we managed to have surviving embryos, they didn't go throughout the whole metamorphosis and became adult, but they survived for several hours, was using naked DNA. So we are now working several different methods of transfection to try to improve this transference. So I'll now go to the talk about some of the challenges that we see ahead. I have like 5 topics that I believe are important for us to briefly discuss. I'll start first with the technological risk, which I believe is, it can be divided in 2 parts. And now I will start quoting Tony Nolan in the meeting that Jean convened and the Foundation for the National Health Institutes organized last November in Brussels, that we were discussing, you know, like all the concerns that populations have about releasing a gene driving in the environment. And he said, well, my main concern is not that it's going to escape and destroy the world. It's actually if it's going to work or not to control the mosquito. So we share this understanding that our biggest concern is not if we are going to be able to build it or not, is if once built, if it's going to work to control the mussel or not. But still, it's a big challenge, which is another aspect of people claiming that we are going to overuse gene driving in species. It's so difficult to build one, so difficult. So I believe that the biggest technological challenge is not, but it's doable, right? So it's not if we are going to, if someone is going to modify the mussel and create a gene drive, is if we, this small group working in Brazil, can, it's up to the task. And I believe with the results that we showed you that we are, and we will continue to work in this direction. So there's always the regulatory risk, right? So we believe we are in a favorable position in related to this, which is that Brazil has a favorable regulatory environment for genetically modified organisms in general. So the Brazilian biosafety law was published in 2005, and the regulatory body, it's called CTN-Bio. It's the National Commission of Biosafety, and they have the authority to regulate and to say if the gene drive is safe, right? So it's a different authority that is going to say if it works, if it's it's going to talk about its efficacy, and that would be the National Environmental Agency. But our first challenge is then to provide all the data for CTN-Bio to give their to talk about its safety. But like I said, the environment, the regulatory framework and environment is favorable. Brazil was the first country to regulate on the self-limiting mosquitoes. It was also the first one to approve the GMO salmon for consumption. So it's a friendly country, which doesn't mean it's going to be easy. So I have another quote from Simon Warner that was the outtake, one of the outtake scientists, that when we talked in Brazil, he said, wow, good luck on presenting a gene drive to CTN-Bio, because a self-limiting organism was really, really challenging. I cannot imagine how it's going to be for a gene drive. And so Gutenberg Souza is from the Brazilian Ministry of Science and Technology, and we have been, and also a member of the National Biosafety Commission. We have been talking closely to them, but the problem is that a regulatory agency cannot work on a hypothesis. I cannot tell them, oh, this mussel is going to be like this and like that, and they can, tell me anything. So we have to get the mussel ready, and then they are going to issue an opinion. And well, then we have to finish building the mussel and see what are going to be. It's hard to anticipate the regulatory challenges. Of course, there's a social risk, right? And maybe this is, I believe this audience knows maybe even more than we do about the social risk, because the perception of the malaria gene drive is, because it's so connected with humans and health, it's maybe more challenging than the golden mussel problem. So when I talk about this to people, I usually try to show that, yes, there is bias and there is prejudice. And the first challenge, it's actually to get people to listen to you without the prejudice. And this short video, I don't know if you know it, what's a GMO? It's a kind of caricature of this prejudice, right? So like a reporter go on the street and ask people whether they consume GMOs or not, whether they like it or not, and they all complain and say no. And then when he asks what GMO stands for, and no one knows actually what GMO stands for. And the only person in the video that knows what GMO stands for is the one that has no problem with it. And I love this image from Matthew Potts. Of course, he's making also, a joke after an article on Nature a couple of years ago showed that there was no evidence of any deaths from GMO consumption by livestock over 30 years of livestock being fed with GMO feds. So this image show like the zero all over the world, zero deaths, zero casualties caused by GMO consumption. But I remember these two sentences. I was in the World Social Science Forum in 2018 discussing gene drive, and there are so many social scientists with so many concerns, and I said, people, you have no idea of how difficult it is to make a gene drive. And I believe that driving is more dangerous than gene driving. But that's just my opinion, to which Joe Husband said, look, even if the risk is low, you have to understand that there is risk and there's the perception of risk, and you cannot ignore the perception of risk from people. So I understand we need to work on reducing this asymmetry of information. I believe that prejudice is caused by this asymmetry of information. And what we have been trying to do is to talk openly to anyone who wants to talk to us about the gene drive, to publish a manifesto in our website, and to have as many opportunities, to create as many opportunities to communicate with the companies, with the traditional populations, with the riverine populations living in the margins of the reservoir, and with the regulators, with the environmental agency, with everyone. Well, this project is funded by the hydroelectric power plants. They have a lot of pressure on them. Not only they are actually, somehow they are painted as the guilty ones, but they are really, they are as victims as anyone else in the reservoir, right? It's just that authorities, including the public attorney and the environmental agents, believe that the power plants which goes under the assumption that they could do something about it, which they can't. And that's why they are now funding this project. It's like, well, this is something that we can do. There's nothing we can do to control the mussel today, but let's then help develop a project that maybe can control in the future. But there's still a big question mark related to whose responsibility it is and who is going to actually buy and pay for the genetic control when and if it becomes a reality. So it's still a really open question. Nobody knows. I don't see anyone volunteering to be the one, having these expenses. So this is one of the big challenges that we have ahead, is to identify a client for this product. And the financial risk is the risk if we are going to be able to raise all the money that we need to move forward with the project, that based on the benchmarks for other species, we believe would be around $10 to $20 million to further develop it. And one of the challenges in Brazil is that the scientific granting agencies don't have this amount of money, and especially to put in, maybe they don't have it all and they don't have to put in one project. And there are no biotech VCs in Brazil that could do that. So the VC community in Brazil is even mature, but for for like Series B, Series C, and in different kinds of businesses, not in biotech. So Brazil has a flourishing fintech community and VC community, entrepreneurial and VC community, but not in biotech. So even if we have, in the opinion of some people, the opportunity to become a unicorn, if we manage to solve this problem, no VCs we are not on the radar of any VCs, and that makes a challenge to raise this money. But we have the commitment of the power plants, of the hydroelectric power plants, to keep funding. And last not least, I want to show you the team. We were in the top, among the top 3 biotech companies in Brazil in 2022. So this is the day we were celebrating. I want special thanks to Juliana Americo, the CTO, the one that really knows about all that is building the construct of the gene drive, Daniele and Yasmin, that are the molecular biologists on the bench building it, João, the bioinformatician, here in the middle, and here on the lower left corner, Fabio and Pedro, that made the magic of reproducing the mussel in the lab. So if some of you can recognize here on the top, the only one not wearing a shirt is Scott Fahrenkrug, that is the founder and was the CEO and CTO of Recombinetics. He managed to genetically modify pigs, cattle, and release several products for livestock. And that is now a partner. And he moved to Brazil, was tired of retirement, and decided to join us in these and in other biotech challenges. That's it. Thank you so much for your attention. I'll be happy to address any questions that you have.
Thanks a lot, Mauro, and really appreciate the talk, and I'm sure the audience did too. Let me just instruct the audience and encourage the audience, one, to ask questions, and you can do that 2 ways now. One, you can chat them in the chat, and we can read them. Alternatively, at this point in in the presentation, we'd be happy to recognize you if you raise your hand, and we can unmute you. You can ask your question orally. So we do have some questions, and I'll start to those in a second, but I just wanted to ask about, first of all, I appreciate the technical challenges that you're facing with non-conventional systems. What I wanted to know was, one of the things I wanted to know from a technical point of view was, were there other mollusk analog systems that might be useful sort of as mollusk models in the lab that would at least help you, assess, maybe get some ideas about promoter functions and construct functionalities and things like that before you actually were able to move it into the golden mussel?
Yeah. No.
No.
So
Okay.
well, there are, of course, the Biofilaria snail. So there is some research in the Biofilaria snail because of its schistosomosis. There was, and there's the zebra mussel. So we published the golden mussel genome soon before the zebra mussel genome came out, and then the quagga mussel genome came out. And what we are doing now is, like, we have a quarterly meeting between all these groups working in mussels to try to control them. Even though each group has a different strategy, we are sharing data, experiences, because in the end, it doesn't matter which technology we use. It's about getting, controlling the mussel population. So, like, even though I'm in love with the solution of the gene drive, I have to, as an entrepreneur and a scientist, I have to be also in love with the problem. And most important for us is to solve the problem. Doesn't matter with which technology.
Yeah, great. So I'm going to go to some of the questions that we have in the chat. Simon Lilico asked a question, actually a couple questions here sort of embedded in here, 3 questions actually. So how well conserved is DMRT1 in bivalves? What's the risk of spread of the gene drive to non-target species? And is horizontal transfer a concern? So a couple things there to address.
So it's well conserved. I would have to reach out to Juliana or João if you want, like, some more specific information on identity and other stuff, but it's very well conserved, and that's why we manage even to find it. So it's very similar in the worm, and in the mosquito and everything. About the risk of spread, we can't say anything yet because we didn't start to do experiments in the lab. So, but all the assumptions that we have are the assumptions for the mosquito, so that the risk is low. But we don't have any empirical evidence of that in the lab because we just managed to have these first genetically modified organisms in the lab. So, yeah, horizontal transfer is a concern. I think maybe I answered this already, saying, like, our biggest concern is it going to work? There's always the risk, and we are going to develop this based on every best practice that exists for the mosquito. We trust that CTN Bio is highly competent to judge on the safety of the gene drive, and we will trust their judgment if it's safe or not. So what we are doing is to build that. And again, the highest risk is that actually the mussel is going to get rid of the gene drive construct, right? Or that we have some kind of modification that makes it useless, and we are working on that to prevent this kind of modification that could eliminate the gene drive functionality. So yeah, I don't have a specific answer based with data for the mussel.
Great. Great. So I'm going to keep going on some of the questions chatted in. Again, let me say that anybody in the audience who'd like to add a discussion point or ask a question, contribute to the discussion, you can do that also by raising your hand, and we'll be happy to recognize you and unmute you. So let's see. Wayne Hunter has a question. Are you using the regulatory definition of eradicate, which remove from a defined area?
Yes. I'm not sure if you saw eradicate in any point of the presentation. We are trying not to use this word. We are aiming for controlling, which is, we don't believe we can eradicate. Like these reservoirs are huge. Like, of course, if you think about, like Africa and the mosquito, that's big, it's huge too. But we believe that, even to avoid all the discussion on should we eradicate the species in an environment, we believe that we want to control. So the aiming is to be able to control that species in that environment, which means keeping them, keeping the population in low density, so they don't clog the pipes of the power electric plants. They don't attach to other animals, to other bivalves, and kill them. They don't attach to the ship hulls that leads to several different problems. There's a lot of corrosion related to the attachment of the mussel. Even concrete is corroded by mussels attached on it. So imagine the pillars of the bridges in the reservoir. So the idea is not to have this 150,000 mussels per square meter especially in the artificial substrates.
Where else is the golden mussel an invasive species and a problem?
Well, actually in China. So it's curious, China is very big, and we just recently, and I say recent, kind of 5 years ago, we've been working with the golden mussel since 2005. And a lot of the literature on the golden mussel was in Chinese, so very difficult to access. But in 2015 or 16, a Chinese researcher, Mengzhen Sheng, she published a review of all the literature on the golden mussel in China. She published it in English, and that was, it was great because we could see that it was already an invasive species in China. So it's original of the Pearl River basin and has spread through the Yangtze River basin and the Yellow River. So now you have reports of the golden mussel in Japan, in Korea, and in Thailand. So all around China region. Curiously, no, but if you look on the map, it's this stripe of the same latitude. So and it's funny because you have a stripe of in the northern hemisphere for the zebra and the quagga mussel, and a stripe close to the equator, close to the equator of the golden mussel in the southern hemisphere.
Interesting. Yeah, it sounds like you need to find some good collaborators over there who can tap into some of the resources over there as well. Anyway,
we have
yeah. Okay. Go ahead.
We did. Sorry, we did population genetic survey in 5 reservoirs in Brazil, because we wanted to know if the populations were different. Then that, of course, would impact the development of the target gene. And no, there's no evidence that the populations in Brazil are different, that you have different populations. Now our next step is to compare this with China, because if this solution is, if we can develop this solution, there would be no reason to apply it in China, where it's causing a lot of financial and ecological damages as well. So now we want to do this population genetic survey between Brazil and China and we are already on the way to do that, to compare the populations.
Ivan Ramashamna has a question. He's a senior scientist at CABI, located in Nairobi. So he has 2 questions. He says, the Golden Mussel is featured in the CABI compendium, and I'm pleased to see this being brought to life with your talk. I just read a recent published paper that economic losses from biological invasions were of a similar magnitude to natural hazards. Yeah, I saw that too, Ivan. To the tune of $1.2 trillion. Have you done some studies to actually quantify the economic losses from this species, like to fishing and the hydroelectric plants and so on?
Yes, there, but not with scientific rigor. It's really difficult to get this information, really, because if you go to a power plant, they don't really quantify how many hours of maintenance is dedicated to the mussel problem. So when they raise those grids and they have to do it from time to time anyway, they don't separate. Okay, I spent like 10 hours, scrapping mussels from the grid, or I don't know, putting oil and lubricants in some parts. So it's difficult to estimate precisely how much of the maintenance costs are associated with the mussel. A further challenge is because of the business model of the electric sector in Brazil. So the hydroelectric power plants are not paid by the amount of energy that they produce. They are paid by the availability to provide energy to the grid, right? So it's difficult. It would be easier to measure loss of profit due to a turbine that is stopped because of maintenance if you just calculated, megawatts hour. But when it's the availability of producing to the grid, it's harder to make the calculations. So we have a number that we came very conservative value. So like it's a low estimate of $120 million a year for the hydroelectric power plants in Brazil. And these numbers of $1 billion in losses, it also includes the cost of fighting the mussel in US. I'm familiar with these numbers for US. There are even bigger numbers for control of different species in Europe, but they are also, extrapolations from point data. So we have to look at them carefully, especially if you want to use this as a market number, right? So we don't know. We don't know. I believe it's around this number. But if you put a lot of things together, a lot of, layer 1, layer 2, layer 3 damages directly to the power plants, I don't believe it gets to this number.
Yeah. He has a second question. how compatible is using genetic control of golden mussels with other biocontrol methods, such as other predatory fish? How do you plan to go about the risk assessment to establish the impact on predatory fish if they consume the gene drive golden mussel?
So these protocols are part of the regulatory requirements of the requirements to apply for a regulatory permit, for a permit to, for example, test the mussel in the wild. So we have a list of tests that are required by CTN-Bio to apply for these permits. And I believe, I'm sure, I know this kind of test is one of them. So what we can do is kind of genotype fish after you fed them with genetically modified mussels, and do analysis, do genetic analysis on the fish in different species that are being raised in the same environment. So it's microcosmos, mesocosmos experiments before we get a permit to go on field trials.
Good, thanks. I'm going to inject one of my questions in here in between. What's the sex ratio of male to female mussels in the wild? Do you know that?
We know that's a very good question because it actually changes. So because they don't have sex chromosomes, we have a whole, so Fabio's PhD is now on this topic, is which genes are activated to determine sex, if they're going to be males or female, and how these genes are expressed throughout the life of the mussel, because we have evidence that they can reverse sex. They can change sex after they are adults. So we have data from surveys in the field showing that throughout the year in the same bank of mussels, they go from 3 males to 1 to 3 to 3 to 1. Okay, so it should be 1 to 1, but it can change.
So I guess that might impact how you model that, right? If
Yes.
Distorting sex ratios can be compensated by sex reversals. So
yes.
Anyway, interesting.
So
interesting.
There's evidence in oyster banks that temperature and food availability can impact the sex ratio. And like I said, we have a PhD thesis being developed on the topic now because it's extremely important.
Yeah. Fred Russell Kumo says, excellent work. Do you think the perception towards your work would be different if the mussel species you are working on was native, not invasive?
Yes. So I think the perception is better because it's invasive. If we were trying to control, not to use the word eradicate, on a native species, it would be more challenging, at least from the perception point of view. Because it is a big problem. So people acknowledge the big problem. So when those aquaculture farms, when they go get the mussels out of the nets, many times they do it on the coast, in the land, and you have these mountains of dead mussels just rotting with a terrible smell. Inside the power plants, you have areas that you have the turbine. I don't know if you are all familiar with the power plant, but you have the turbine here, the water comes from here, and then it has a kind of carousel for the water to leave after they pass the turbine. This carousel chamber is completely taken with golden mussel. And to enter there to give maintenance, simply the levels of ammonia are so high that they are deadly. And People have to first discontaminate the ammonia so they can enter, so people can enter to give maintenance. So it's a big problem for the industry. It's a big environmental problem. And of course, for, I don't know, for me the Rio de Janeiro state is not infested. So yes, there could be people in the country that never heard of the mussel, but if you are in an infested area, you know, it is a big problem.
Yep.
And of course, preventing it from getting to the Amazon is the number one it's the number one priority in the agenda of the environmental authority in Brazil.
Good. I see your colleague Joao has chatted in a message. I'll just say it orally here so everybody else in the audience can know. He says more details on the DMRT gene characterization and comparative analysis is coming up in the genome assembly paper. So
stay tuned.
Stay tuned, exactly. So Rebecca Castro asked a question, and I'm glad she asked this question because I had the same question in mind. In the preliminary research, did you find that the mussels had any effect on other animal populations in the Pantanal and Amazon regions, like otters, birds, et cetera? So, you know, so the implication was in the video, at least, that there was an impact on biodiversity. So is there evidence there?
I'm reading the question to see if I fully understand. So did you find that the mussels had any effect on other animal population in the Pantanal? So there's a lot of research about the impact of the invasive mussels in the food chain, like I showed the fish, right? And a broader impact is the change that they make in the water, in the water quality. So there's evidence from research in Argentina, from Argentine researchers, that they consume all the plankton and they release a lot of ammonia, which leads to blooms of cyanobacteria. Right? So all this change, but I'm not aware of long-term whole ecosystem studies to measure the impact of the golden mussel. So I'm familiar with, let's say, these point studies measuring this. Regarding the toxicity of the genetically modified mussel on the other populations, like I said, this is a topic. So our idea is to follow the lead and the regulatory framework of CTN-Bio to assess the impact on different species. If they include birds, I sincerely don't know, and I don't think it does.
Mm-hmm. Yeah, I think it would be interesting.
But we would be happy. We would be happy if anyone wants to execute these risk assessments. We'd be happy to. We still don't have the mussels, but we would be happy to provide them for people to execute different risk assessment experiments.
Yeah. Any strong evidence you had on impacts on biodiversity could be a real powerful driver for subsequent for the intervention down the road, for sure. Yeah. So okay.
I even believe, Dave, that I flipped this idea just recently. I don't think we should start testing. This may be controversial, okay, but I don't think we should start testing the mussel once we get all the regulatory approvals and once we are ready to go and test, and once it has been tested and proven safe and effective, I think we should start in places where, not to reduce the population of the infested basins, but to prevent the infestation of the pristine rivers, which includes the Amazon River. because it will get there. It's just a matter of time. So we could go, like I can explain a bit more why it hasn't get there yet. There's probably a control of, so the flooding control in the Pantanal watershed, it's probably controlling the population there, and reduced the number of mussels that escapes to the Amazon River basin. So this is an area where the Plata River, where the I don't know how to say in English, where the rivers are born, nascentes in Portuguese would be, in like 200 kilometers, so 100 miles from of distance, you have the rivers that start and go form the Amazon River, and the rivers that start and go form the La Plata River. And, it's a 2-hour drive. So if you are in a fishing competition or a recreative fisheries, and you have your boat, and you go and you're fishing in this infested river, then you take the boat, put in your car, and take it to a pristine river that actually connects with the Amazon River basin. This happens every day. Okay, so it's just a matter of time. So I believe that we are in a rush to develop this, to be able to prevent this inevitable infestation, try to avoid to get there in the first place.
Yeah. Just a quick one before we go to the next question. When you showed that fish that had eaten all those mussels, was that fish preying on the mussels because it was a food source, or was that fish, a healthy fish that's what they do? They eat mussels?
Yes.
Or was that a negative impact on the fish?
Oh, it's just an abundant food resource. Yeah. Okay. Right. So if you are in the environment, you have all this food resource, let's prey on it. So it's even challenging when people say, there are no natural predators. That's not true. A lot of fish, they eat the mussel, but it's not good to their health. So they can't make the judgment call that it's going to be difficult to digest the mussel. Or maybe the food the food item that they are used to prey is not available anymore, so they would prey on the mussel. I'm not familiar with research in this field, like trying to understand why one food item and not the other.
Yep. Okay, I've got a couple more questions. Ivan Mugenia has two, actually. So what's causing the population boom of the mussels, and any environmental impact assessments done for animals coexisting with it? So I think we covered some of that, but the boom part, you might want to just talk about why they're so prevalent.
I don't have a good answer. I would, these waters are eutrophicated, so there's a lot of foods available for them to eat. So they and actually that was the challenge of reproducing them in the lab. It's not about reproducing them, it's about feeding them. We did some experiments to try to understand what was their limit, how much they would eat, and there's no limit to it. We spoil the water with food before they get tired of filtrating. They are simply going to eat everything that you put there. And it's hard to get a diet that will keep them happy in the lab, so they spawn. And I would guess that the natural amounts of organic matter in these Brazilian rivers and reservoirs are continuous source of food supply these animals that are genetically prepared to reproduce a lot. So they reproduce 3-4 times a year in Brazilian, in the Brazilian temperature. If you go to Argentina, already lower latitudes and colder waters, you have one spawning season in Brazil. It's almost continuous. So like you have of course, during the summer, have a higher chance of finding the gametes in the environment and spawning populations, but they reproduce a lot.
Okay, and his final question was there any, this is from Ivan Mugenia, was there any information on the prevalence of mutations in the host gene where the construct is placed?
Yeah, no. This is our next, this is one of the, when we continue developing the genetic program for the mussel, of course, this target analysis it's super important and necessary. So we haven't found these genetic the population genetics experiment survey that we did was looking at the whole genome, right? So like markers in the whole genome. We do have data on SNPs in the target gene, but we need to increase the number of organisms and population that are analyzed to understand if it's going to be a problem or not. But we do have some transcriptome data that we are from different organisms that can allow us to improve this. But this is an ongoing research on the mutations on the host gene.
Great. I'm going to, before we sort of wrap things up here, since we're out of, we don't have any more questions at the moment, I just want to make those in the audience who are still in the audience aware that this webinar series will actually continue next week. We'll have our next webinar in this series, and we will have Joanna Tanos speaking about possible gene drive control of a pathogenic fungus that affects some trees that are significant economically. So hopefully that everybody will be able to join us then as this series continues. So again, an unconventional system for the potential application of these technologies. It'll be very interesting to hear what she has to say. Mauro, I don't see any more questions in our chat, and I don't see any hands raised. And so at this point, I'm going to say thanks a lot for participating in the webinar series. It was really interesting, and I think it was super informative, and really appreciated the whole story that goes into applying these technologies. And your comment about this is not easy is really true. And it's such a misconception in the popular press that, oh, we can gene edit anything, and it's just sort of conflation. Well, they don't even know they're conflating it, but it's a misinterpretation. While the enzyme is highly efficient, it's not easy to get it in the right spot at the right time. And yes. I think that's what makes it hard.
Inside the cell.
Exactly.
Well, we can go on and on about the challenges.
Yeah, so the enzyme itself is a great enzyme, right? And yes. It's really, really pretty efficient at doing what it's supposed to do. But getting it to where it wants to go, that's where you want it to go, is the big challenge. And I think the work that you described today really illustrates that there's a lot of other things that need to be solved before these before this technology solutions can actually be implemented or even constructed and tested. So anyway, wish you a lot of luck, and I hope we stay in touch for sure.
Definitely.
Really, really interesting, and thanks again.
Thank you all, and follow us on social networks, and you have my email there. we would be happy to follow up on the discussion. Thank you.
Wonderful. Thanks to everybody in the audience. And with that, I'm going to say have a nice rest of your day and hope to see you back next week. Bye-bye.